Welcome to the WTR Healthcare Happenings podcast. I'm your host, Tim Gerdeman, Vice Chair and Co-founder and Chief Marketing Officer of Water Tower Research. In today's podcast episode, I'm being joined by Dr. Marvin Hausman, Co-founder and Chief Scientific Officer of Ludwig Enterprises. Also joining us is my Water Tower Research Equity Research colleague, Robert Sassoon. Ludwig Enterprises, OTC ticker symbol LUDG, is at the forefront of a new era of medicine with its proprietary novel mRNA genomic technology platform, which has the potential to measure genetic biomarkers for inflammatory-driven diseases, including but not limited to heart disease and cancer, the world's two most chronic diseases. So without further ado, let me welcome Marvin and Robert to today's podcast.
Well, it's a pleasure to be invited. I'm excited about the podcast.
Great to be here too, and I'm really looking forward to it.
Today's podcast will cover the topic which many consider to be the holy grail of healthcare. That is early disease detection and preventative medicine, and we will discuss where Ludwig fits into those areas. Before I hand it over to Robert to ask some questions, though, I think it's always interesting for our audience to learn about the individual behind the role. So with that in mind, Marvin, can you give us a summary of your professional background and what brought you to the very interesting world of genomics?
Sure, I've had a very exciting life, and it's a pleasure to share it with you. And my whole mantra is to leave a legacy for healthcare, and that's what I want to do now as I've gotten older. My career started, I've been over 50 years in the pharmaceutical and business world. My career started when I became an MD at New York University School of Medicine. I'm still on the board of governors of the Alumni Association, the Langone, now it's the Langone Medical Center. I then had my residency in general surgery at Mount Sinai Hospital. I was drafted, believe it or not, into the Marine Corps in 1968, and then suddenly my orders were changed, and I was invited to go to NIH. I was a lieutenant commander, and I'm a research associate, and that's when I started working on the immune system and macrophages. Before that, I was invited to UCLA, and I helped develop part of the HLA system, the histocompatibility system, in the lab of Dr. Paul Terasaki, who is the guru of histocompatibility, and that's how you type people for transplants, whether it's kidney, heart, lung, liver. So I've been working on that my whole life. So from NIH, I then was invited to UCLA, where I became a board certified urologic surgeon. I subspecialized in pediatric kidney surgery and also in transplantation. I then went on to co-found, I just love business and medicine. And despite the fact that I had a very active practice, I consider it at one time the largest pediatric surgery practice in urology at Cedars-Sinai Medical Center, the big hospital. I still was able to co-found a company called Medco Research. And we then ended up going on the New York Stock Exchange. We were acquired by King Pharmaceuticals, and now we're a division of Pfizer. And this company I founded is in Raleigh, Durham, North Carolina, and is part of Pfizer. The next company I co-founded was Exonics, and we became one of the leading Alzheimer companies. We acquired technology from NYU. We acquired technology from the National Institute on Aging, and then we actually moved a whole department from NYU to Serono in Geneva. And Serono now is a part of Merck of Germany, and Exonics now is part of Raptor Pharmaceuticals. We initially went to Torrey Pines and then merged into Raptor. I then decided to look at alternative medicine. And what is life about in terms of cancer and Parkinson's disease and Alzheimer's disease? And I was approached by people at Michael J. Fox Foundation to do a study. And I came up with the concept of using nutritional products from mushrooms with the help of a gentleman who's now emeritus professor at Penn State, Dr. Robert Beelman. And we developed a substance called ergothioneine with vitamin D, high levels of vitamin D. And Michael J. Fox sponsored the study, and we showed that we could modulate an mRNA product or peptide in the brain of animals with Parkinson's disease. Now, this was a double-blind, placebo-controlled trial. These animals were created at Stanford. They were genetically modified to have all the symptoms of human Parkinson's disease. And I couldn't believe this. And then when I moved to Florida, I was approached by a gentleman called Frank Magliochetti to form a company called Designer Genomics. And he said, let's look at your theories on mRNA. What is the role of mRNA? I said, it's music to my ears because mRNA is the language of DNA. How does DNA speak? How do we become who we are? Now, for example, you take the DNA from your mother, the DNA from your father, yet you are a unique individual. It had to express itself, and it expresses itself through mRNA and microRNA. And you differentiate into who you are. And I said, well, maybe this is how cancers differentiate. They're not any different than we are. They grow inside of us. And maybe there's a language coming out of cancer that we can identify. So I said, Frank, why don't you introduce me? His wife has a lab called Genetic Institute of America, and she has a relationship with Thermo Fisher, the largest genetic company in the world. And they introduced me to some key players there. And I gave them 80 hits, and they were able to sequence 48 of my mRNA suggestions. And then we set up a study through Genetic Institute of America across the United States. At that time, she had probably 10 multi-centers. Now she has, I think, 80. And we accumulated over 3,000 specimens. And we went ahead and started to analyze them for mRNA and using NGS, next gene sequencing. And lo and behold, we identified 6 major mRNA players that were involved in breast cancer. We studied breast cancer, colon cancer, and also breast, bladder, and bladder. And so we were able, and so we just presented, and it was published in the ASCO Journal in 2025, our cocktail of 6 mRNA genes that are involved in the screening for breast cancer.
That's a remarkable introduction to include a very robust professional background, Marvin. And I have to say, as an aside or digression, as a former member of the Ohio Army National Guard and Maryland Air National Guard myself, thank you for your early service to the country. And over to you, Robert.
Thank you,
Yeah,
Tim.
I have a very sympathetic leaning to veterans. Initially, I was obviously in the Marine Corps, then they transferred me to the Navy and then to the Public Health Service. The same, and I'll just make a statement. We can discuss it on another podcast. What we're working on, the language of mRNA, is also the language of PTSD. Mm-hmm. I told you Parkinson's disease. These are central nervous system inflammatory reactions. Post-traumatic stress disorder is not a genetic problem. Okay? And that's what our military is subject to, our first responders. And what we're talking about today could be diagnostic and a screening for PTSD and determining the severity of PTSD. So sorry to digress for a moment, but you touch a chord in my body. I'm very sympathetic to veterans.
Same here. And Robert and I have actually discussed this topic on prior podcasts, so we will definitely circle back to get you back on that topic, which is near and dear to my heart. So, Robert?
Yeah, you're coming to Florida. 2 years ago, I presented in Tallahassee on PTSD, and I'd be happy to share that with you when you move to Florida.
That would be great. Robert?
Hello, Marvin. We've witnessed quite a few advances in disease treatment, like heart disease and oncology over the last couple of decades. So, do you think advances made in fixing diseases, or at least improving the quality of life of disease sufferers, has been matched by advancements in early disease detection?
Oh, absolutely. I think we're on the cutting edge of a whole new millennium. We have to move away from the previous formulas that we used in medicine. We have to start thinking outside the box. Um, we need, and people are having a difficult time being their own advocates in healthcare. And they can't always get to see the doctor. They get to see, no disrespect, they're well trained nurse practitioners, physician assistants. It's very hard even to get your own blood tests. So we need to establish an advocacy program, personalized medicine to move into this new millennium. And that's what I would like to do. And I think you're seeing that with all the telehealth, a company called Hims and Hers. I was listening to a program where they said the telehealth market is $150 billion. That's unremarkable. And Hims controls 40% of that market, and they foresee the telehealth and personalized medicine market going to over $1 trillion in the next 10 years. So we're on the brink of a major cutting edge change in medicine.
Right. So I mean, going back to the early disease detection and preventative screening, what are the breakthroughs that's particularly stand out for you that you've seen advance in the last in recent years?
You know, it's fascinating. When I was at NIH in 1968, 69, 70, 71, um, my mentor asked me to work on macrophages. These are the killer cells for cancer and bacteria. And I was able to identify what in the immune system causes that macrophage to go to a cancer cell. What's very interesting now is I created this mRNA program using cheek cells and saliva. You swab, simply non-invasive swabbing. You don't need a biopsy. You don't need blood tests. You don't need a mammogram. And we analyze the 48 genes, mRNA, that are in that cheek swab, and they're stable. You have to develop the technology, of course, because mRNA can be unstable compared to DNA, but there are preservatives that could stabilize it. And what's fascinating is now I see articles coming out that the body communicates with the mouth. The mouth is not a mouthpiece just for your voice. It seems to be a mouthpiece for cancer. That cancer is distal to the mouth. The breast is distal to the mouth. Why are we finding it in the mRNA as a diagnostic screen? Colon, pancreatic. I just saw an article, a meta-analysis, where they go around and they look at all the articles that are out there. They don't do the research. They do the research on the people who have done their research using artificial intelligence, area under the curve, false positive, false negatives. And I was just, oh, I just, but anyway, I was looking for the article. I thought I had it on my other screen. But what was fascinating, 85% positive findings in saliva for cancers that are outside the mouth. And one of the major ones is pancreatic cancer. That's a death knell. That's a
That's amazing.
silent killer. Ovarian cancer, silent killer. Even breast. I mean, women need to become their advocates. And we're on the pathway to allow women to take care of themselves. Just a simple cheek swab. And we'll see if you have a red light, yellow light, or green light. Green light is a zone where you should, that's nice, you're taking care of yourself. Yellow light is your personalized inflammatory index came into the yellow zone. Maybe you should go and have an exam with a GP or whatever doctor you like. And red zone is maybe you should go have a mammogram or maybe an MRI. And this is where medicine is moving. And it's exciting to be there.
That's amazing. So actually taking that further, can you maybe talk about Ludwig's mRNA genomic technology platform? And there are maybe some podcast listeners here who are not scientists. So maybe if you can give us a 101 on what mRNA is, and you've really explained maybe to some extent, maybe you can develop
Yeah,
further
no.
what makes your platform a good disease detection system. And its advantages and disadvantages when you compared with other biomarkers. And there's been a lot of there's been an explosion of research in recent years on biomarkers for various indications. So maybe you can just delve into that a little bit.
Okay, so we have DNA. DNA is a fixed molecule. You know about it. It goes back to the Human Genome Project under when Clinton was president. And all of this has emigrated from that original project, where they identified and were able to look at the entire human genome of animals and humans, mammals, all organisms, and they now can look at the entire genome. Well, DNA is fixed, but DNA sends a message. DNA is in the nucleus of the cell. What produces lipids? What produces proteins? Well, you have a manufacturing area in every cell. And the message from DNA to that manufacturing factory is mRNA. So when cancer occurs, it's like a wound that doesn't heal. It's inside your body. And my theory is, and other people now think of it also, that it tricks the body into saying, I'm a wound. Come heal me. Come send me proteins. Come send me lipids. Let me grow and let me then kill you. I'm sorry to discuss it that way. And that was what excited me about looking at that language. What is happening in a normal breast cancer cell in a control with these 48 mRNA genes, and what's happening when it develops cancer in the 48 mRNA genes? And that's what we have now identified. But we couldn't do with standard statistical analysis. For example, when we did we have 48 mRNA genes. And then we take a cheek swab and we have to study all 48. Think about that. 48 in 69 patients that we selected, 30 controls. The permutations and combinations are 12.2 million. It's staggering. And a good colleague of mine is now at Nike. He was at Intel when I worked with him initially on the Michael J. Fox Foundation Parkinson study. Kyle Ambert is brilliant. And he developed an algorithm using machine learning called an assemblage process. And he was able to identify these 6 out of 12.2 million permutations and combinations. So we filed a patent. In that patent, we didn't take all 12.2 million. We took 3,300 that had above an AUC, an area under the curve of 0.84 or 0.85. And that is your standard area under the curve, because that brings in your false positives and your false negatives. And I don't want to bore the audience with an F1 index, which is what you look at in machine learning, artificial intelligence. I'm just looking at called an NPV, negative predictive value. And on NPV, we're going to be showing to the world when we launch our test now is 90%. I think no other company has that degree of NPV.
Right.
Screening for breast cancer, and we want to go next into pancreatic cancer. So I think we're sitting on a very unique occurrence, and you could have all the other tests that are out there. We're not undermining GRAIL or undermining Cologuard or undermining mammogram. We just want the person to say, I need to get some further testing. This is a screening test. This is not a definite diagnostic, but it allows a patient to get up in the morning and think that maybe I can look at myself and make sure I'm healthy. And I think that's what mRNA represents.
Right. So can you actually comment on, you take that for a little further and comment on the screening accuracy and reliability of the platform?
And I sent you the data from ASCO. So we
Right.
have better data now. The data from ASCO. Let me explain what you do if we have enough time. Have you ever driven in Waymo, the new car, the autonomous car in Chicago? You
I,
call it.
I have not, but I'm certainly keenly aware of the technology.
It's amazing. I know I was in Los Angeles before the fire. My two of my children are doctors. My son is an anesthesiologist, went to Northwestern. My daughter went to Yale and then Penn, and she's a maternal fetal medicine doctor. And my younger son is in real estate, but his wife does search engine optimization. Two out of the three lost their homes in Pacific Palisades fire. So just before the fire, I was there, and we went to dinner. And I said to my daughter's boyfriend, I said, "What's that car with the, it looks like a radar thing on top?" He said, "Oh, that's Waymo." He says, "You want to go in one?" I said, "No, no, 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 I'm not going in any self-driving car." He says, "Come on." So he picked up his phone and he called it and he told it exactly where he is. 8 minutes later, the car pulls in. This thing on top is circling and going around. We get in, there's no driver. It was a freaky experience. And we're weaving in and out of traffic. It's stopping at the lights. It's obviously sees a car next to it and knows how to slow down and go behind it if it wants to make a right turn. So I said to Kyle Amber, I said, tell me, are you a Waymo? Are you taking mRNA? And he says, I didn't think of it that way, but yes. He says, because I took the 69, and this is the data I can't show you right now, but we will. I took the 69 patients that we presented at ASCO and the 30 controls, non-cancer patients, and I used them to train my algorithm. So he used it was a training of the algorithm. So he was similar to what Waymo does in terms of the radar. It looks around. That car is trained to identify things.
Right.
Then, once he did the training, he went to other specimens we have, which we have not presented to the public. And we took, I think, 38 breast cancer, 38 non-cancer controls, and they were not given a diagnosis for the machine learning algorithm. The machine learning algorithm didn't know if they were normal or cancerous, but all it did was know the mRNA data. And it came out with an MPV of almost 0.90. Think about that.
Well,
This is
amazing.
the Waymo of the future.
Right.
And that people are, negating artificial intelligence. We got to control it. We got to let it go. We got to use it for healthcare. We got to save lives with artificial intelligence and machine learning. And that's what this company is.
Great. I have to come add throw in another question because not everybody is a great fan of mRNA technology. And yesterday, of course, RFK Jr., the Health and Human Services Secretary, um, culled announced that he's going to cull 22 mRNA vaccine projects, basically because he claims that mRNA technology is too risky. Now, he's only talking about vaccines in this case. I don't know what his opinions are about
Oh, you didn't
technology
may not want to
elsewhere,
hear
but
all my
I mean,
opinions.
what are
By
your
the way,
reactions to his? You're a very experienced scientist and physician. What are your reactions to his skepticism
well,
on
skepticism is good. Plato, who I haven't met, said, "You never know the answer, you only approach the answer." And I think certain assumptions have been made in medicine. Now, we really have to differentiate. Now, I'm an immunologist. I worked on the HLA system to type people for transplants way back in the 60s. So I'm very familiar with all the technology, the vaccines. Polio is a totally different subject. Polio, was revolutionary. Salk and the Salk vaccine and the other gentleman also. Um, they're making a vaccine to work through your immune system. They're not using mRNA. Okay. COVID is an mRNA virus. COVID then stimulated Pfizer and Moderna and the German company to produce this mRNA, and they call it a vaccine. The problem's there, and I'm not familiar with which ones he's pulling, but the problem's there, which I'm very concerned about. And I'm not saying people shouldn't use it, but be aware that they have not definitively proven that the mRNA that they're giving you is not combining into your normal genome.
Right.
Yeah, why? My son almost died. Sorry to bring this up. Who now his house burned down after vaccination. He developed myocarditis. He's young. And why are these young people developing these heart problems after vaccination? Why? I mean, his heart function went from 450 down to 10. Ejection fraction, incompatible with life. So he went to the hospital, he was there for months. They put him on medication. Thank God he's back at about 40. He's back, not back to normal.
Okay.
But I think we have a lot to look at. And I can't comment on what he did yesterday because I don't know the exact ones he pulled. But we have to look. Data should support us. If we don't have the data, and you know as well as I do, and I have to be careful on this statement, that the pharmaceutical companies control the FDA.
Right.
All the money they have, most of the money comes from pharma. When you submit a drug, you have to pay.
Right.
To play. Except now there are divisions like the orphan drug division. And I'm now working in pancreatic cancer and glioblastoma. That's another project I'm working on as a consultant in another company. And I put in an orphan drug application for glioblastoma for mRNA in an exosome. And so I'm now, if I get that approved, that gives you the benefit of exclusivity in that area if you ever get the drug approved. So the FDA is really moving away and allowing creativity and smaller companies to come into the game. But for a long time recently, we couldn't come into the game.
Robert, I would be remiss if I didn't comment here that Marvin's candor is very refreshing.
It's great. Yeah. Anyway, I think I just wanted to bring that point up because
It's a touchy subject. I
a
mean,
touchy subject, and
I'm
I'm glad
happy.
you,
I'm happy.
you.
Listen, I'm not saying that anything is bad. I didn't say that. I'm saying we have to look at the data. Data doesn't lie. And the jury's still out on mRNA. What's going to
Right.
happen 10 years from now? What's going to happen a year from now? We have a lot to learn. And the research we're doing now could be used there
Right.
because long COVID, I'm sorry to digress for a minute, occurs. And that's when the symptoms, you have no more virus, but you have all these other symptoms related to the initial viral infection.
Right.
mRNA that we're developing has the potential to identify patients for long COVID. We just don't have the funds to do that. But maybe in the future, when we uplist and we raise money, we can look at long COVID. We can look at PTSD. Why does someone commit suicide?
Right.
Committing suicide is not a genetic disorder. It's a hyperexcitatory state that occurs on the nerve endings in the brain, and people do things that are strange. So we
Right.
have a lot to learn, and I think the fact that we're looking and identifying and searching, I think is very positive.
Right. But there's also one thing that it's for use for actual treatment as opposed to early detection. So that's something that we probably have to separate out.
Yeah,
So
I'm doing that. That's why I
yeah,
applied for the orphan drug application. And we can discuss that in another podcast.
Yeah, I'd love to discuss, but
No,
in the interest of time, I just want to finish up with these questions. So, where are you actually in terms of the platform's development, your platform's development, and what timeline are you looking at for commercialization? And finally, how large do you see the market opportunity for mRNA biomarkers?
well, let's take the last one first. It's vast. It's unreal. I mean, I don't know how the number of people who would need this is astronomical. To go backwards, the first thing we have to do is to validate the test, which we've just done. So we hired an outside third party to come in, just like you go to Quest or LabCorp and you get a potassium or sodium, they have to have that test validated. So we just finished validating our test versus PCR. I won't go into all those details. That we just completed. We are now ready to launch. So I think, God willing, sorry to talk about God or whatever force you believe in, that we may launch within the next 2 months. I would like to launch by October, but I'm not the marketing guy, and I'm the chief science officer. But
How large do
we
you
have
see?
gurus in the marketing area that are working out. So we've identified the swab that accumulates the mRNA. It's made by a company called Copan in Italy. They have a division in the US, and you have to have a certain amount of our mRNA, and we've acquired that technology. You need 20 to 40 nanograms. I won't bore you with that. We're there. And then we've created the marketing box that when a patient calls up, then you need digital backup. You need back office. You have to get the result to the patient and to the doctor. And we're designing the portals for that. We've been working on that for the last 6 to 8 months. We're really close. We should be launching within the next 2 months if all goes well.
Great. And how large do you see is the market opportunity for that platform?
I stay away from that. I mean, the whole world is your oyster. I mean, cancer is a killer. I mean, just I'll give you one example. India, I think they now have the largest population in the world, right? Despite their diet, the highest incidence of diabetes is in India.
Right.
And the highest level of breast cancer is in the diabetic patients in India. You know how big that market is? I mean, how can I sit here and just froth at the mouth and tell you that we're going to be $1 billion company? But yeah, it is $1 billion market for us initially, but
That's
we have to
great.
get there.
Yeah. Well, I was just throwing in that it is definitely looks like it's going to be a huge market if you're and hopefully you'll be able to get some success in that. So thanks very
But there are
much for
baby
all of that.
steps. You have to do baby steps before you walk, before you
Sure.
run.
Yeah, for sure.
And that's what we're doing. I'm a surgeon. You verify before you do something in surgery.
Right. Okay.
We're verifying to make sure that we make no mistakes.
Well, thank you for all that insight, Marvin, and I'm going to pass it back to Tim.
Thanks, Robert. Ironically, Marvin said at one point, I'm not going to bore you. There was virtually nothing about this conversation that was boring. It was absolutely fascinating to me. So thanks for joining us today, Dr. Marvin Hausman, co-founder and chief scientific officer of Ludwig Enterprises, OTC ticker symbol LUDG. And thanks to my equity research colleague, Robert Sassoon, for also joining us today. And last but not least, a special thanks to the WTR producer and editor of the podcast, Joe Brunetto. Thank you for listening. Don't forget to subscribe, as well as visiting www.Water Tower Research to stay up to speed on the company's small cap written research reports, podcasts, fireside chats, and conference schedules. We will see you next time for another edition of the WTR Healthcare Happenings podcast.